Everything is fine. I'm just trying to install the presentation because we still don't have it.
Okay, we see, yes, this is screen, yes.
So yes, you see everything. You might see everything now. And we're just trying to install some like this. Yeah, so everything is fine.
Yes, perfect.
So, okay.
Okay, the floor is yours. Sergey, Shityakov. How is correct with V at the end of double F? It's like German style, Shityakov.
Yeah, this is fine. I mean, I actually, for me, it's hard to talk about it because I don't really see the people. So,
okay,
I mean,
majority they will look the presentation online.
I see.
And then someone connected in Zoom, and some people I will record your presentation, and the group leader. If you allow us, okay, I will send the group leader. They know what is your focus.
Okay, so you see also you see my picture, right?
Yes.
Okay, everything is fine, so it should work now.
Okay, the floor is yours. We are recording, and okay, happy to hear more about your research. Okay,
sure. Yeah, so you're welcome to ask me any questions. Yeah, so okay, she's not here. Anna is not. Okay, sorry, some ambient noise. Okay, let me just explain what we.
Okay, we somehow didn't hear you. It happened something with the sound. Can you switch on the microphone? I think it's somehow starts automatically switched off all microphone. I think it's you just need to click on Zoom that will start audio. I think we switched off somewhere.
So hopefully we'll have it. Yeah.
Now it works.
Yeah, so I got this back, but yeah, it's okay now. Hello.
Yes, yes, we can hear you.
Yeah, sure, but you don't see the picture, right?
We don't see the picture now.
Yeah.
I think you need to switch on your camera.
Because I don't know, this is just not working. Yeah. Okay.
Now we can hear you and we see you.
Yeah. This is not easy because okay. try. I have to rewrite. Yeah. Okay. Looks good.
It's perfect.
Yeah. Okay. So but you don't really see anything. You really see the picture, right?
We see the pictures. Okay, now not any longer.
Yeah. Okay. Okay. Today I want to give the presentation, which is titled "The Rational Drug Design and Testing of Lead and Test Substances with as well as cyclodextrin formulated drugs." My name is Sergey Shityakov, and I'm a leading researcher at the University of Würzburg. So as you can see, Würzburg University, University Hospital. I mean, we have different facilities, and here you see in the lower part of the screen, you get different facilities, different buildings. And in one of these buildings I'm located, where the laboratories, we are doing some experiments. On the other hand, we are also having some collaborations, so we have to use some other locations in a different laboratories. Yes, and so first of all, I would like to give an introduction. It's all about what we are doing, what my lab is doing, and what we are focusing on. So most of the projects are related to different substances. Cyclodextrin, some saccharides, small drug-like molecules. As you can see on the upper part of the slide, there are some substances, even some bacteria and viruses. So it's more like, so let's say, some as exposome, let's say, and this exposome has some effects on the human body. So we try to investigate how all the substances of the molecules, also some toxins, either environmental toxins or synthesized toxins can have some effects on the human body, because we are focusing on biomedicine. And in particular, we are focusing on neurosubstances and also some drug delivery vectors, such as carbon nanotubes, cyclodextrin formulated substances. I was also doing my PhD in virology, so then we tried to investigate the viruses, neurotropic viruses, and the part of the virus, like viral capsids, and how this can be used in terms of the transfer the genes and some drug-like molecules. So on the left part of the slide, you see that we have some achievements. The citations, you see the growth of citations, and I got some H factor, H index, and currently it's 14, but close to 17 maybe right now. So far I have 70 publications. Some publications I even made to publish in the highest impact factor journals like Nature Communications. I'm also very active in doing reviews. And so far we're also doing a lot of peer reviews. And yeah, this was what we are doing so far. And we have also a facility. Some facility includes the laboratory equipment, and some facilities they include some theoretical equipment. I'm not putting all the information, but we got a lot of equipment, which is starting from routine cell culturing and finishing with some electron microscopy and scanning electron microscopy. And you can see that we are doing a lot of sets of experiments in silico, in vitro, in vivo, ex vivo clinical studies, which represents experiments on the chemistry of substances, concerning various chemical characterization. Also some experiments are designed to evaluate the substances, measuring the cytotoxic potential and mutagenicity. Yeah. So what we've done so far, and we are working on the cell-based models, where we're trying to set up some assays to measure some properties, some dynamic properties of the substances, like the permeation of the substances through different barriers. It's fine. The sound is okay. Can you hear me? Hello?
Yes, we can hear you all. It's perfect. We see your presentation and can hear you.
Okay,
I just switch off the microphone, not to disturb.
Okay. Yeah, so here I'm just putting some information that what we are doing. We are harvesting some cells, and these cells, after some weeks of incubation, some days or weeks of incubation, they're ready to do the experiments for the permeation of the drugs, of the substances. And the permeation means that the substance gradually permeated through the monolayer of
What
the
type of
cellular.
cells are these?
Pardon?
What type of cells are these?
These are the endothelial cells. They are from the brain, and they're immortalized using some viruses. On the other hand, we're also using some primary cell lines like neurons. So it can be also can be used. Depends on the model. If you're focusing on 2D model, which is easier, we can do the primary immortalized cell lines. If it's 3D model, which is more complicated, so I would just try to use also the primary cell lines, which was primarily prepared from the animals. And you also see the graph. This graph tells some information on the transendothelial resistance when we measuring when we're trying to measure to evaluate the tightness of the barrier. And if the tightness is fine, then we will do the experiment. Sometimes also when we temper the cell lines with some substances, you can see the differences in the tightness of the cell, of the monolayer, and the differences in the transendothelial resistance as shown here. For instance, if you have a treatment with some drugs like dexamethasone and with some other substances like dexamethasone antagonists, you see the differences in the tightness of the cells. On the other hand, we got also the investigation of the cellular tightness in terms of the expression of some proteins. And these are proteins primarily some tight junctional proteins, for instance, aclodins, clodins, catecherins, which are highly expressed in the barrier level. And if you got a high expression of this kind of proteins, you also have a higher barrier properties. So we also do the Western blot for this kind of proteins, so we're analyzing Western blots. The other projects is screenings. Some screening would be screening of some drug-like substances, screening some substances which are formulated with some chemical structures like polymers or oligomers, for instance, oligosaccharides as cyclodextrins. As here I present in the results, where we have also the crystal structure of some transporters which are highly expressed at the BBB, as for instance the BBB choline transporter, which is active at the BBB and can be used as a drug delivery vector to enhance the permeation to the brain. As you can see at the graph, you have a very distinct clustering of different substances. Some of them, they are clustered in a group which is active, and some of them they're clustered in a group which are inactives. And this clustering is done by measuring the free energy of binding, which is binding affinity of the molecule to this transporter. This was also published a couple of years ago in High Impact Factor Journal. The other screening was done measuring the multi-drug resistance, which was known to be for the PGP, which is P-glycoprotein transporter. And this P-glycoprotein transporter is a very huge molecule, transmembrane, and is known to be highly active at the blood-brain barrier level, and it's in a flux transporter. It's an ATP cassette, a flux transporter. As it is well known to expel all the substances from the site of action. Therefore, we have to measure the flux of the drugs, whether the drugs are known to be inhibitor or to be a substrate or to be inhibitor for this transporter. Together with this, we are measuring in parallel. We are also measuring the viability of the drugs, the cells, using the drugs, whether the cells are survive after the treatment after their treatment after 24 hours or 48 hours of exposure, drug exposure. As you can see, when you're measuring the cellular availability after cellular viability after 24 hours and 48 hours, so far we didn't really find any toxicity using our drug delivery vectors, either formulated or not formulated with some drugs. So, for instance, we are using fitzdextran, which is kind of polymer. On the other hand, you see that we get measured the single wall carbon nanotube in comparison to the dimethyl sulfoxide, which is 10% and which is known to be very toxic. On the right part of the slide, we are measuring the molecular representation, the molecular properties of the molecules as a pi stocking, which is involved, for instance, as it is well known for the aromatic substances because they are establishing the interaction, while the pi stocking, which is involved in a PGP interaction, for instance, some fullerenes and carbon nanotubes, uses this kind of mechanisms to interact with the PGP. And the other experiment was done using also some drug delivery vectors together with some reporter molecules of fluorescein thioisocyanate molecule, which is known for instance, to give some fluorescence, and you can actually measure the signal of the cells. And this experiment was done quite some time ago, and we measured the tightness of the cells. Again, measuring some tight junctional proteins, transendothelial resistance, and I also did some confocal microscopy. I was trying to investigate the localization and colocalization of this kind of drug delivery vector. As you can see, there is a 3D plot where the localization and colocalization of the signal can be found, and therefore you can discriminate. You can also measure the aggregation of this drug delivery vector. Okay, the next is also a similar project was performed where we did also crystallography, X-ray crystallography, and NMR studies on some substances, where we tried to investigate the interaction of some dopamine derivatives or levodopa molecules with some cyclodextrin dimers. As you can see here, some molecules they tend to go to the interior of the cells, and some molecules tend to go to the outside of the cells. We're also measuring the toxicity levels for these substances, and together with the in vivo studies, where we got.
Just can I ask the question concerning this, for example, particular work, from the point of computation of these systems, how you are choosing the object? Okay, what is shown here in gray, and the small molecules are drugs. Am I correct? Or?
You mean the brain and the blood and brain? The graph which shows the blood and brain represent the ratio, right? I'm not really sure.
No, we are seeing now your picture, and then picture there is some channel which is shown in gray. What is this channel about? And what then there is a pink molecule inside, or pink something inside.
Right. Yes.
And
Yes.
around molecules, okay, I'm not a theoretician, but I'm just asking you from the point of view, you are in majority doing the computation, or you're also doing the cell work?
Yeah, you're right.
Someone.
Yes, you're talking about the 3D reconstruction, right? The picture
Yes.
shows the 3D reconstruction. Okay, I'll just explain it to you. This is the NMR model of the cyclodextrins, and we're trying to, because we are using the monomer, also dimer, sometimes it creates some kind of a polymer. And this polymer is known to have an interaction with some drug-like molecules, like dopamine, for instance. And here is shown that dopamine tends to bind to the interior of the molecule, to the interior of the cyclodextrin dimer here. So at first we tried to just to show that this kind of small spheres, they represent the binding site of the molecule, of the dimer, where the complexation is taking place. Yeah. And the second would be the dopamine and levodopa, they're relatively small. So they're going to the interior of the molecule, to the dimer molecule. Okay. So the next is we try to measure the interaction of the derivatives. As you can see, the derivatives, they tend to aggregate at the exterior of the dimer, because there is a phenomenon of the chemical branching. This was written in my paper, why we try to investigate the mechanism, why we have this kind of phenomenon. And some molecule without this chemical branching, they tend to go inside of the catalytic channel. It's not a catalytic channel; it's a kind of binding channel, if you want. But the part of the molecule still appears outside of the dimer. So this is what we found so far. We also using the experimental techniques, some techniques like NMR, and we also doing some in silico studies, so together with experimental theoretical results. And the 3D plot shows the clustering. So we also measuring some surfaces, which are giving us the information that this chemical branching also provides the clustering of different molecules. So as you can see, we get 3 clusters. So I'm not going into detail, but we are measuring also some constants, and so, as you can see, the Gibbs free energy of binding is also measured. Yeah, so this is the result of our experiment. And then we did also the pharmacokinetics, pharmacodynamics, but I'm not showing it here. So on the right side of the slide, we got the cytotoxicity, and we got the in vivo experiments when I'm injecting the cyclodextrin complex into the mice. Then I do the operation. I'm harvesting the organs, and these organs can be measured. The concentration, or the drug concentration, can be measured directly into the tissue, in the tissue. And this is the result. So from this graph, one could also deduce the partitioning coefficient, for instance, log BB. Yeah. As you can see, different complexes are also tend to have different permeation potentials and log BB. So I'm not putting the log BBs here, but you might also notice that the concentration in the blood in the brain is different, and therefore it might also affect it. It will affect the BB permeation. So the next project was just published recently in the high ACS Journal of Medicinal Chemistry, which is the highest impact journal in the medicinal chemistry. And we did here a PK/PD modeling of neuropharmaceuticals. We tried to try to measure pharmacokinetics, pharmacodynamics. In the upper part of the slide is made. I did the experiment. We did actually not me, but also some of my assistants did, and I supervised the experiments. And the upper part of the slide, you see that you have pharmacodynamics, and the lower slide of the lower part of the slide you get pharmacokinetics, but the substances are different. So only the upper part of the slide was published, and the lower part was just the data from some pharmaceutical companies. So they also do some collaborations with us, and this was done using some protein ligand interaction systems, like for instance propofol and human serum albumin. And in fact, we measure different albumin. It's not just human, but also canine, bovine, rat, rabbit, and sheep. And from this, one can also calculate so-called unbound concentration 50, or IC50, but it's not really IC50 because we use the cell-free system or animal-free system. And here on the right part of the slide, you see the interaction, also the crystal structure. You see the interaction of the propofol with the human serum albumin. The human serum albumin has 2 binding sites, which makes the interaction even more complicated. And these binding sites are involved in the interaction. And I'm also using the conservation and so-called conservation analysis. As you can see, there are some residues which are more conserved. There are some residues which are less conserved, and the ligand is also shown to establish some H bonds with the receptor. Yeah, and of course the lower part of the slide shows the pharmacokinetics of this. It's a standard pharmacokinetics curve for the slide. The only thing is the left part of the graph is shown for the IV injection, and the right part of the graph is shown for a porous application. So it's a little bit, you might probably notice a little decay, little kind of delay of the concentration, and after some time you got an exponential increase in the concentration in order to reach the C max, and then you get the exponential decrease or decay of the concentration. Okay, next is all about the chemical characterization. We got different methods. They include scanning electron microscopy, transmission electron microscopy, X-ray powder diffraction, also together with in vitro and in vivo experiments. So here first we did some scanning electron microscopy, then I did a reconstruction to measure the granularity of and the porosity of the structure. And this was done in parallel with the characterization of whether the structure is crystalline or amorphous. As you can see here, the experiment, you see that the graph represents that our structure is amorphous here. And we also did, as always, we also did the measurement of toxicity, and we did the in vivo permeation, the blood-brain barrier permeation. Here is actually the permeation in a monolayer. As you can see, there are 2 different curves for the permeation of the drug. And this drug, on the right side, the figure A is shown for propofol, and the figure B, where the concentration is shown for sevoflurane. The classical curve for the permeation is of course the linear regression. Although for the sevoflurane, the linear regression is not the case, because this drug is known to be volatile. Therefore, after some time, when you increase the incubation time, you also increase the possibility for the drug to go into the gas phase. Therefore, you're losing some concentration in the experiment. And this also represents the decline of in the concentration. Okay. So, yeah, so this is the, okay, so I did something, yeah. Sorry, it's okay, right? Or...
Yes, but we again see your presentation that it's now in the mode of...
Okay, right? It's back.
Is
it back?
Yes, it's back.
Okay, so it's back. So the next project was done to evaluate the cytotoxicity of different cyclodextrins and also mutagenicity. So we did, as I already explained you, we did a cell-based assay for cytotoxicity, which is shown here, but the assay is a little bit different. We did microscopy of the cells, and then we converted the microscopy, quantitative microscopy to the qualitative microscopy to the quantitative measurements, where you see that the different cyclodextrins represent different toxicity against the BBB endothelial cells. And together with the COMET assay, the COMET assay is known for the mutagenicity, which is the mechanism is that if you address the substance to the cell, if the substance is mutagenic, then it will create some kind of fragmentation of the DNA. This fragmentation of the DNA can be also detected. The highest fragmentation of the DNA is known to be as a positive control from the H2O2. As you can see, this was almost up to 15%. And when we try to use the negative control, then normally it's 5%. So when we address the H2O2, it increases up to 15-20%. And here, the experiment, you see that our substances have no mutagenetic effect on the cells. Sometimes we also do an AIMS test, but our tests tend to be more precise because we are using mammalian cells. And of course, the outcome of the experiment is the fact that cyclodextrins are known to extract some lipids from some lipid molecules like cholesterol from the lipid membranes. And this extraction might lead to the cellular death, apoptotic events in the cells. And therefore, you see that also the interaction, we check the interaction of the cyclodextrin with lipid molecules. In this case, this is cholesterol molecule. And of course, the binding affinity of the cholesterol to the cyclodextrin can be precisely correlated to the cytotoxic profiles of these drug delivery vectors. Yeah, so the next is a little bit more information where I was doing during my PhD. This was done in the using virological assays when I was doing a lot of cloning, trying to create some gene expression vectors when I can express, for instance, genes in non-dividing cells, and the idea was to infect non-dividing cells such as neurons. Therefore, I tried to utilize the mechanism in lentiviruses, which are known to infect HIV, which are known to infect non-dividing cells, and therefore, but the only concern is the safety. The safety of lentiviruses is not that good because they are also known to promote cancer and malignant tumors. Therefore, we try to use some vectors, some viruses, which are known to be very, very safe, but not very efficient in terms of infection of this kind of cells, neurons, and some cells which are restricted in different stages of the cell cycle. here I was doing some experiments and I restricted the cell cycles. As you can see, the results quite interesting. So I get some vectors which are able to infect non-dividing cells, and I got also different clones. I got different viral vectors which are generated. So I'm not going into details what kind of molecular elements I have. This might be very complicated. It involves a lot of retrovirology here and the mechanism how retroviruses are going into the cells. So the idea is that they're creating some kind of pre-integration complex in order to go into the nucleus, and some other viruses don't really have this kind of complexes. Therefore, at that time, we thought if we transfer some genetic elements to some other viruses, then we can also rescue these pre-integration complexes in order to infect non-divided cells. So this was the idea. Yeah. And this might help us to create some kind of some genes which can be expressed directly in the nervous tissue. We can modify the behavioral aspects of the animal. We can modify some disease-related profiles. For instance, here, as you can see, different pathways. This is from my other paper, which was published quite some time ago, and we were trying to investigate the effect of HIV virus to trigger some pathogenic profiles like dementia, like HIV-induced dementia in the patients, in HIV patients. Yeah. So next was also done in our lab. We're also doing a lot of experiments on the pain, and here is just the result where we're evaluating the drug delivery vectors. The same, they are cyclodextrin-based drug delivery vectors in terms of decrease the pain level in the rodents. So here, this experiment we are doing using some behavioral assays like pulse pressure assay, some chemical characterization, as you can see in the upper part of the slide. There is a figure A, where we're measuring the degree of substitution and how this degree of substitution would link to a solubility constants using different fractions of cyclodextrins, and how this would correlate to interact with some prostaglandins. These prostaglandins are known to be the pain mediators. The hypothesis was if we try to increase the affinity of drug delivery vectors, in this case cyclodextrin-based vectors, to prostaglandins, so we can also reduce the pain level in the animal. Therefore, these drug delivery vectors can be also implemented together with some painkillers. So this was the idea of the experiment. Actually, the results are quite nice. We found that prostaglandins are able to interact with the beta-cyclodextrin, especially with the randomized methylated beta-cyclodextrins. And the effect is that in vivo experiments, they will decrease the pain in animals. Here, just a couple of words, what we were doing in collaboration. I'm not going into details. This was published in Nature.com, and we tried to use the different substances to in a model where we are trying to investigate the diseases, which is tuberous sclerosis, and we did on tuberous sclerosis murine model. This disease is kind of neurodegenerative disease, which you get like patches also in the brain, and some kind of some diseases also confined in humans. And here we did the experiments. We used the patch clamping to measure the action potential in the cells, and how the drugs, the newly synthesized drugs, like prototype drugs, not treated drugs, let's say, prototypic drugs, drug-like molecules or lead compounds. So how these lead compounds would also affect the action potential in this kind of model. And this was amazing that we found this drug really can be very beneficial for this kind of disease, and we were able to reduce the amount of seizures in the animal. So next, I'm sometimes also doing systems biology using some machine learning, and here I'm just a couple of words. This was just submitted to a high impact factor journal just last week. I just submitted it last week. And here we did some measurements. This was measured in a population of healthy people, which were taken some medications, especially one group was taken the PUFA, which is known for polyunsaturated fatty acids, which is abbreviated for polyunsaturated fatty acids like APA, DHA, especially some specific fatty acids. And the other was taken placebo. As you can see, you got also nice results that, in fact, I was able to cluster these two groups and discriminate after I was implementing the machine learning effect. The machine learning is the very powerful technique which will give us actually the future possibility to discriminate and to model the biological, biomedical data and biomedical phenomena like gene expression profiles, like microarray data, big data. Sometimes you also have information from the binding data. So this kind of data can be also implemented in the machine learning. So here I created the big data, which we obtained from the microarray assays. So some couple of words on the future project. Here we are also doing on some rational drug design and discovery in Alzheimer's disease. In Alzheimer's disease, you might know that there are some two targets. One is the tau protein, and the other is beta amyloid beta peptide. So in the upper part of the slide, there is a tau protein, and the lower part of the slide, there is a amyloid beta peptide. And we try to, right now we're searching for a drug which might be beneficial to disrupt the complex, to formation of the plaques in these diseases, in this particular disease. And so here you can see the tangles, and our hypothesis right now is whether we can actually stop the tangles from a tangle formation by using specific drug-like molecules. Yeah. So this is the methodology so far. We are implementing the X-ray crystallography. As you can see here, we got like kind of protocol, what we're gonna do. Actually, we already obtained the crystal. You can see in some crystal on the left side of the slide. On the right side of the slide, you have the diffraction map of the crystal. And from this diffraction map, one can obtain the X-ray first, the electron density map. And from this electron density map, you can actually obtain the 3D structure and do the analysis. Yeah, so the future plans, what are the future plans, development and potential contribution? So first of all, we are still We are still going through many projects, many collaborations in drug design, discovery, bioinformatics, computational chemistry, organic chemistry, infochemistry, biomedicine, vascular research. This is multi-international, multidisciplinary projects, and we have a lot of collaborators. So some of our collaborators are shown here, but they are not all. Right now we have more like 50 collaborators from different countries. So the next would be to publish, of course, the results and the patent, the application. We also have some patents. I'm not showing here, but we have. We're focusing on very high impact factor journals like Nature, ACS journals. Usually we're publishing Nature, ACS journals, or cerebral cortex. The impact factor is usually more than 10, sometimes it's 20. So, okay, it might be also 7 ACS, like some ACS journals, they have 7, 8. I'm also doing a lot of supervision and deep PhD supervision, supervising a lot of students, attracting third-party funds like BMBF, DFG grants in Germany, Synergy, Horizon 2020, teaching. I do a lot of teaching, delivering a lot of seminars and lectures, and also establishing new collaborations and establishing and publishing together with my colleagues. So these are the future plans, and of course we're expanding, and yeah. So thank you for your attention, and I'm happy if you get any questions.
Thank you very much. Okay, the questions. I have a lot